All right, recording is running. It's my great honor to welcome Professor Murat Eren to today's HIDA Lecture at Data. And I've promised to keep it really short and not talk too much about his CV and his research. But I want to just say that Murat is a computer scientist and microbial ecologist. And I think before joining Helmholtz Institute for Functional Marine Biodiversity just this year, Murat was an assistant professor at the University of Chicago for a couple of years. And when coming to Germany earlier this year, Murat brought part of his laboratory, part of his team to Germany. And they continue to still work on the establishment of the intersection between actually marine microbiology and computer sciences, which is a perfect fit to more data, by the way. And while doing so, Murat is focusing a lot of the creation of advanced open source software, right? And practicing open and reproducible science practices. And this, again, fits perfectly into what HIDA is trying to promote anyways. Today, Murat is going to be talking about high-resolution integrated multi-omics and some of the challenges that are related to that. And maybe he's going to help us all to make some sense of the extremely complex environmental data sets that I think not only he, but many of the participants today are actually bringing home from their field trips. So, as I said, we're going to have time for questions after the lecture. However, if you have urgent questions, please feel free to interrupt Murat at any time. Unless you have an urgent question, please keep your mic muted. And without much further ado, Murat, the floor is yours. Feel free to share your screen and go ahead.
Thank you very much. Thanks for the invitation, and thank you all very much for being here, for your time. Let me start by sharing my screen. Here we go. I hope you can all see this. And can you also see when I move my mouse around the screen? No, not quite. Okay, well, I'll keep that in mind. All right, let's start. Thanks a lot for the introduction. I'm a computer science by training, as Eno mentioned, and I am applying some of my understanding from computer science, data visualization, and so on to marine microbial ecology. So I was told by Eno that most of the audience will likely not have a strong background in biology or microbiology. And one of the aims of HIDA lectures I learned is to make them accessible to a broader audience. So while my aim was to perhaps too extensively talk about some exciting challenges in today's data-enabled microbiology, with that understanding, I decided to also include a relatively longer background to discuss some context, with the hope that I can offer some new insights into microbial life for at least some of you here. And my ultimate goal here is to get at least some of the early career data researchers among us excited about potential opportunities that could benefit from their skills if it haven't crossed their minds that this could be an option for them. Okay, so my talk is 3 parts. I will start by talking about microbes a little bit and try to make a case for microbes. Why should you be interested in microbes, actually? And then I'll talk about evolution just a little bit. Hopefully, within 20 minutes, I'll do my best to fit everything into 45 minutes here today and focus on some new and exciting challenges for lovers of life and data. And then I'll talk about future a little bit, how what I think else is coming towards us if we have time. So let's start with microbes. Why do we care about microbes, or what microbes bring on the table in life sciences? So probably most of you already know this, that maybe you don't think about this very often, but microbes indeed were the first life forms on Earth. And when Earth is formed as an uninhabitable hot mess, about 4.6 billion years ago, the universe has already been around for about 9 billion years. And it took another billion years for the first life form that could actually use the energy from the sun to create biomass to emerge. These organisms were doing anexogenic photosynthesis, which means there were no free oxygen on the planet yet. Their metabolic byproducts did not include oxygen. And 700 million years later than those guys, the first oxygen producers start to emerge on the planet. The waste product of their respiration was free oxygen, which was chemically captured by other elements that were abundant in the environment, such as iron. And when dissolved iron oxidized with oxygen, it sank to the bottom of the oceans. And we know about this today because it left behind these astonishing layers of iron formation. So that's how important that particular transformation on this planet. Microbes work under these conditions for about another 400 million years. And about 2.3 billion years ago, the atmosphere finally could start accumulating oxygen. This is an event that played a fundamental role in shaping the diversity of life on Earth, because with the availability of oxygen, more energy-demanding forms of life could emerge, because now the oxidation of organic matter to generate energy, the form of respiration we all on this call also depend upon, was now possible. And finally, 500 million years ago, the first terrestrial plants emerged, and 300 million after that, the first mammals emerged. Only 10 million years ago, the branch of life that includes us started to shape. And there we were after that. And 400 years ago, we finally observed for the first time who was truly responsible for all this. So when you think about the timescales here, it really is interesting how recent it is that our discovery of microbes. When you think about other sciences, they were thinking about all the things microbes influence without knowing their existence. So microbes, the mighty architects of our planets, are also extremely abundant and diverse. Their abundance is very easy to communicate. I always find it less challenging than their diversity. For instance, I tell people that there are 40 million bacterial cells in a gram of soil. 1 million of them occur in a milliliter of seawater. And then I ask them to consider the amount of large bodies of water on Earth and then just extrapolate the number, which is about trillion droplets of water there is on Earth, which is septillion, if you prefer. And then you add a few zeros to that number to start just picturing in your mind the number of microbial cells that live in our oceans and seas. And each of you, in fact, harbor approximately 1 trillion microbial cells that live in your gastrointestinal tract largely, but also oral cavity, urogenitary tract, and so on. And so you are made of about 1 trillion eukaryotic cells. When you think about that, the number even becomes more astonishing, in my opinion. So yeah, microbial life is extremely abundant and diverse, as in the degree of variation of among life forms, when I say diversity in this context. So the explanation of that is a little more challenging, but I will give it a try. I guess we can all agree that life is one of the most interesting phenomena in the observable universe. And being challenged by the astonishing diversity and beauty of the members of life, humans must have been asking questions and wondering about systematics of life quite early. Especially one question that has, I guess, forever been very popular: how many different kinds of living beings are out there? Must be an ancient question, in fact, but we will never know how far this goes back. But there is one name that manages to make its way all the way today, is the first person to attempt to classify all living things in a systematical fashion. And that's Aristotle, who is seen here in Raphael's nice fresco, telling Plato to calm down. He comes out with the first systematic attempt to classify life into meaningful groups. Of course, these groups are not as meaningful as one would hope, even from those times, perhaps. But even though the resolution was very low, this was the first recorded attempt towards this kind of goal that dominated biology for a long time. And you go forward about 2,000 years, and we had more detailed representations of the diversity of life as these trees and bifurcating things and so on. But at this point, all efforts to classify life did not take into consideration microbes. none of these trees around 1800s had no microbes in them. Although people knew they existed, they had no idea about how to attach them to these trees. We want to understand diversity of life, but where do microbes fit? But while Ernst Haeckel, one of the pictures I showed in the previous page, was busy putting man at the top of the tree of life in Jena, Robert Koch, who was only a few hours away from Berlin, actually, was becoming the father of microbiology by bringing to the laboratory environment some of the very first representatives of this microbial members of life. People had just started talking about all the time on TikTok, of 1800s. And what you see here is the first photograph of isolated bacteria, which I discovered recently that this exists. I was very excited, but this thing has only 34 citations. So, talk about high impact work, not making a big sound. But despite that, the fact that now we were bringing these microbes into the lab environment and so on, it was still extremely difficult to make sense of their diversity, given that all efforts to classify life relied upon morphology, and people couldn't add microbes to these trees due to the lack of morphological features in the microbial world. For instance, these are two very distinct microbes that I will detail a little further in a couple of slides, but they look extremely similar. So biology has now solved this problem to a large degree, thanks to significant advances that took place during the last few decades in sequencing technologies. And thus, we can actually compute a tree of life that includes all domains of life, independent of morphology, which often recapitulates morphology, but without having to rely on morphology. And now I'll just show you one of those trees, just to help you appreciate how distinct these two similar-looking microbes are, and how you are actually no more than a carbon copy of a fish as far as the diversity of life is concerned. So here, I'm starting with a portion of the tree of life that represent all mammals, which includes things that, span across the entire cute animals section of your perception of nature. And includes us and so on. But here, all this diversity is represented by 5 branches. The reason for this is because when you try to adjust the resolution of the tree, that's how much space we get at the end of the day from the entire real estate of diversity on life. So just imagine that. And here, all animals are represented on this tree, which includes things that you perhaps not consider as animals, like rotifers or sponges or Toxoplasma gondii and so on and so forth. And here, you are seeing everything in eukaryotic life. This includes not only animals and plants, but also algae and fungi and protozoa, everything you ever saw around and or didn't are represented here. Already, the vast majority of what you see here is microbial, to be honest. So, but it continues to be impressive. This is archaea and eukarya now shown here. Archaea is another domain of life that most of you probably are less familiar with compared to other things, but those are also single-celled organisms. And the rest of the diversity is representing bacterial diversity here on Earth. And so when you look at this tree, this is where this fish is, and this is where you are. And essentially, the genetic distance between the two of you are this much. So, just to put this in context, this is where that microbe, one of those microbial organisms is, and this is where the other one, and this is their distance. So, this is one of the interesting moments that makes you realize that you will never be able to imagine the distance between these two microbes as far as their metabolic activities go, compared to how you are not a fish, essentially. So we have more better trees that are more refined, and in my opinion, there are a lot of philosophical implications of this discovery of where we are. I don't think humans even appreciate this just yet. We haven't truly wrapped our minds around the fact that everyone we know and every living being we can see around us, and the ironical contrast to how we feel entitled about the fact that the planet is ours to reshape, ours to protect, ours to take care of, and so on. So there are some philosophical implications, but microbes are more than the architects of life, or a lesson in our lack of modesty. It is through their astonishing genetic diversity they continue to catalyze chemical reactions that keep Earth a planet that can support complex life, as they play immense roles in the flux and recycling of essential components of chemical components of life, and thereby sustaining their overall bioavailability. So Earth is a closed system, and chemical reactions are very important to recycle some of the components that are used as building blocks of certain life. I hope by now I was able to convince you that one who wishes to understand life as a whole cannot ignore microbes. But perhaps even more critically, at least from a very pragmatic point of view, we can say that to develop a precise understanding of environmental change, we must focus on microbes, as they are the first responders to any change. So in part with this motivation, my group has been trying to understand strategies by which microbes respond and adapt to environmental change. And we have been doing that using a myriad of omics approaches. And this is an immensely data-rich field of microbiology, and extremely exciting to people who appreciate the complexity of life and enjoy or able to work with complex datasets. I can spend an hour, and I'm not even exaggerating that, on every single arrow in this display you see here. But I'm not going to do that, lucky you. But you should feel free to get in touch with me if you have any questions, or if you'd like to discuss these things more, as I always am happy to discuss about make people lose sleep over some very exciting and difficult data problems. So just throwing it out there. And for the remainder of my talk, however, I want to focus on a single area of study that present one of the most advanced data challenges that I have in my life. That is going to bring me to the second half of my talk. Evolution is a topic everyone in life science is very excited about for good reasons. And some of the most fundamental questions for those who study evolution include things like, how does the genomic heterogeneity emerge within environmental populations? How does it derive the partitioning of ecological niches? Or which variants are maintained by neutral versus adaptive processes? So these are some of the really big questions of evolution. The first question, obviously, is to understand how genomic diversity proliferate in individual populations, which, of course, is the sacred process of nature that eventually leads to speciation, and the one that created all the branches of life as we see it, right? Because as the genomic diversity proliferates within a population, it ends up once in a while turning a single population into multiple species, a function of time. The second question is to understand how the environments we study and figure out how they influence and influenced by activities of the ever-changing entities of life. So there is a connection between the diversity at the very low genetic level and environmental responses of life, of course. That's how selection acts upon, after all, on these variants. And the third question, the third question is the holy grail of all these, given its potential implications on medicine and biotechnology and everything else. So we really, really would like to understand which variants are maintained by neutral processes of evolution, and as important as having two different t-shirts and different colors, and which variants are extremely important for survival, like having a head or not having a head in a human body, and so on. So with the increasing availability of environmental data, today we are much closer to addressing at least some aspects of these questions. One strategy that gives us a lot of new opportunities to characterize genetic variability within environmental populations is called metagenomics. I'm not sure if you are familiar with metagenomics, but metagenomics essentially is the direct sequencing of all DNA found in an environment. So you have an environment, you have these large eukaryotic genomes, or I lost my mouse. Or some bacterial genomes or plasmids floating around, or viruses. What you do is you do shotgun sequencing, and as a result of our not quite talented yet sequencing chemistry, you get a lot of metagenomic short reads. So it's almost like aliens come on the planet, and then their alienship is not good enough to sample living organisms, so they chop everything up on the planet and then bring up to the ship. So they work with like individual pieces, and then they try to piece things together. So This direct sequencing of every piece of DNA found in the environment offers us an amazing opportunity as metagenomes enable comprehensive sampling of genetic diversity, because you get everything from everyone in a way if you're deeply sequencing, and that enables some new ways of analyzing the diversity of life. For instance, one of the strategies we use is metagenomic read recruitment. When you have these metagenomic short trees that come from all over the place, right? Let's say every color represents a different species for the sake of the argument now. If you have a representative genome, you can use that as a reference sequence, and through read recruitment, you can identify all the reads that match to this particular genome. It's like if the aliens have a mannequin or a single human that's whole, they can find all the pieces that match the human body in a way. And the beauty is, of course, this is completely de novo, which means even when you don't know the colors, by extension, you don't know where these reads are coming from, essentially. By using the same strategy, you still can't find those that match that one genome. And then using the coverage information, the amount of reads a given genome recruits from the environment, not only one can learn about gene level differences, the coverage of a given genome in a different environment across different environments and so on. So not only you can characterize the biogeography of the population as well as gene distribution patterns in its environmental pangenome, but you can also focus on very subtle things, such as how reads that match to a given genomic context slightly and subtly and systematically diverge from what the reference sequence suggests. So this tells us, of course, about the population heterogeneity in a given environment, given the genome we have to create a framework to understand that. And we do that through single nucleotide variants. People, going back to these big questions, people have been using single nucleotide variants to approximate answers to them. But you may be wondering, how can this extremely simple strategy turns into an extremely data-rich investigation of some of the most fundamental questions in evolution. So now I'll try to demonstrate it, like how do we use this in our learnings into it further through our own studies that maintain the that focused on genetic diversity within populations of SAR11 marine systems. SAR11, I wonder how many of you heard of it, but it's a microbial clade that was discovered about 30 years ago, and it's a free-living marine microbial clade that makes up roughly 30% of all bacterial cells in open surface ocean waters. Just like the way humans are in every continent, populations of SAR11 are in every part of every ocean, imagine. So thanks to its well-representation in oceans, we can actually study population genetics using metagenomes, because when you do metagenomic sequencing, you will get a lot of reads that belong to SAR11 in a way, right? But until recently, ocean metagenomes were quite hard to come by. They were very specific to certain region and so on. But thanks to this heroic efforts by Tara Oceans, this is a multinational project, and thanks to this project, we are drowning in metagenomic data, so to say, from all oceans and seas. So now we have a genome and a set of metagenomes that come from environments in which populations represented by the genome flourishes. We can do the same metagenomic read recruitment and start focusing on things. So now I just want to change gears and talk just a little bit about how do we study genetic heterogeneity within a single population we observe in the environment. So it more or less goes like this and relies on read recruitment, again, as you can imagine. But I'll give an example here using a single gene for convenience, but it's going to be the same for the entire genome. So let's say we have an operating frame, which obviously has a particular sequence. And if we also have metagenomes like Tara Ocean and SAR11 genes in this context, you can imagine from environments in which this population occurs, then we can actually use those sequences in this genome to do read recruitment analysis from different environments. And again, as I said, not only we learn about the coverage of this organism in different environments, but we also can characterize its genetic diversity in a given sample through a single nucleotide variant. So these variants can, of course, help us quantify selective pressures the population experiences in theory across spatial or temporal scales. And people have been doing similar things for about 2 decades now, looking at metagenomes and being able to recover SNVs, single nucleotide variants, and then go from there to understand what is really going on. But SAR11, things got very exciting very quickly when we discovered that the state-of-the-art way of studying population genetics from this point on suffered remarkable shortcomings to contextualize genetic variation as we saw the extent of genetic variation in SAR11. So this is one of the areas that my group has contributed and focused a bit, and I wish to detail them for the next few slides to give you a broader sense of the nature of the data here and ideas that that are out there to work with today. So 2 of the shortcomings we realized in state-of-the-art approaches, I could summarize into one, accurate characterization of synonymous versus non-synonymous nature of genetic variance was very difficult with this increased amount of diversity. And I'll give you an example for that and show you how we kind of went beyond that. And 2, putting these variants into a context to understand evolutionary forces that maintain them was an extremely difficult task to resolve. So what do I mean by the first one? So SAR11 is really a very unique microbe. It doesn't really listen to what people think about how microbial evolution should work and how microbial species should be defined and so on and so forth. When you go to an environment when everyone else looks like this, SAR11 looks like this. So basically, algorithms that worked well to study population genetics of most other organisms do not work for SAR11. Is the immense amount of variation within SAR11 populations make it almost impossible to quantify synonymous and non-synonymous variation accurately. And here's an example why. Let's say you have a codon that encodes for serine in your genomic context. Let's say this is one of the SAR11 genes, and this is one of the codons in it. We can study the variation, and serine has 6 different codons, right? There are 6 different ways to code for serine. Of course, this is a hypothetical example. SAR11 will not have all 6 codons represented in its genome, and that's how life works usually. But let's stick with this toy example to highlight an important point. We can study the variation we find in the environment by aligning these short reads to the genome and looking at individual nucleotide positions. So for instance, here we have A's and T's. So there's some variation in the environment, 50-50. And then even though you observe both A and T in the first nucleotide position of all serine codons, you know that you should also look at the rest of the nucleotides to see if TGT also encodes for serine, and you realize that, oh no, TGT encodes for something else. So probably 50% of the variation I observe here is non-synonymous, essentially changing the amino acid sequence, right? And then you look at the second nucleotide position, you see another single nucleotide variant there, and it goes within C and G, and you look at the list of nucleotides you have, and you see that ACT is not one of the serine codons, and you say, okay, this is probably non-synonymous, but AGT is among serine codons, so that's probably synonymous. So you again maintain the idea that about 50% of variation is non-synonymous in this environment so far. And then let's say the third nucleotide position, there is nothing to look at. So your conclusion is that in this particular environment, based on population, the cells that live in this environment, this population has about 50% non-synonymity at this particular codon position. So, okay, but synonymity is not a property of nucleotides, but codons. So what we did, we decided to focus on the entirety of these metagenomic shorties that fully map to a given codon position by discarding the rest, and then looking at the frequencies of those codons and see which identities they resolve. So for instance, even in this toy example, in fact, 100% of the variation you observe in the environment is non-synonymous. So that's when we realized that, we realized that actually trying to understand processes that maintain genetic heterogeneity in naturally occurring populations using algorithms that are designed to deal with simple problems are, of course, over-challenged when they are thrown on SAR11. So this is an interesting lesson on how initial, I think, observations and computer scientists can inadvertently delay progress in life sciences, because we came out with this. The reason biologists are looking at pileups and not entire codons, even though nucleotides have nothing to it describing synonymity, but codons are, is because some computer scientists somewhere decided that, oh, we need to look at, it's computationally very feasible to look at pileups rather than individual codons in fully aligned reads, and they implemented it like that. And it's been, basically, we'll be using it for the reason of practicality, that tools existed. So, okay, that's another topic we can argue about later, perhaps. So we started using metagenomes in a way where we could actually characterize things a little more carefully. And then we came out with this terms of single codon variants and single amino acid variants, that we could literally count frequencies of different codons and then look at the amino acid identities of those things and so on. And we came out with using a single genome and 0.4 billion reads it recruited from the environment. We had 100,000 single amino acid variants to now understand how environmental factors might be causing evolutionary traits to emerge in sour-living populations in the environment as a function of temperature or whatever. So I want you to just for a second imagine the complexity of this data. What I'm telling you is that there is, imagine this as a table. Let's say every column in this table is one of the codon positions in sour-living genome, and every row in this table is one of the metagenomes, one of the environments we look at. And the cells in this table, what do they show? They show you actually a 20-dimensional vector where you have the frequencies of every single amino acid, and yet not even you can compute distances using Euclidean distance between those frequency vectors, because not every amino acid is identically distinct from the next one. You have hydrophobic one, you have hydrophilic ones, you have positively charged amino acids, and so on. So even within the universe of amino acids, you have a structure that you may want to consider if you want to do a very good job understanding evolutionary processes. So we worked with some colleagues of ours who were interested in actually deep learning, and they kindly worked with us, and we developed a few algorithms, including an auto-clustering layer for deep learning neural nets. And we were able to finally describe that the signal we learn from genomic heterogeneity of these environmental populations show us that their proteome differ between different environments, and these distinct proteomes match to large-scale ocean current temperatures. So, and one of the interesting things we observed that the differences between despite the difference between rather closely related geographical regions, there were crazy similarities between very distant locations. So, for instance, proteome D, I guess you don't see my mouse, sorry, I keep showing things to you, but proteome D are very connecting very distantly located ocean locales based on how proteome converges to generate a similar signal when you look at single amino acid variants. And the striking connection between geographically distinct regions of the oceans based on sour-living prototypes indicate that single amino acid variants are not primarily structured by the global dispersal of water masses, but instead tend to link distant geographical regions with similar environmental conditions, which strongly refutes a neutral proliferation of variants in these genomes. So they are mediated by some sort of selective pressures that require certain amino acids to be fixed in certain regions and not in others, and so on. So therefore, this kind of selection is a strong determinant of sour-living evolution of biogeography. That was our conclusion from these data. And we published that paper with Tom Dalmont, who was a postdoc in my group at the time, and Evan Kiefel, who was a grad student at the time. And so with that, we now had a way to characterize synonymous and non-synonymous nature of genetic variants. But then we also realized something along the way. We realized that single amino acid variants, when you look at those different prototypes that I showed in the previous figure, when you take a random gene from SAR11 genome and look how those variants look like in the context of the 3D structures, we could see that there was an enrichment in cold waters and depletion in warm waters and vice versa and so on. But the main point was that single amino acid variants were not uniformly distributed. Even though even our relatively advanced approaches to this very complex problem compared to practice that have been implemented before inadvertently assumed so. So that was not the case. Then we realized that we need to do something about the proper contextualization of variants to understand evolutionary forces behind them. So I want to now go back to this basics of the approach that use single nucleotide variants that I really like. It's very cool, very smart, but this time I'm going back to it to destroy it. First, I want to give you a few reminders. May be very clear to those of you who study population genetics, but genetic variants found in environments are maintained by neutral as well as adaptive processes. Some variants are simply neutral. You change your t-shirts every day, but other variants, like you put your glasses every day, and compared to other people, most of them don't have glasses or have glasses. Glasses play a more significant role in your fitness than your t-shirt, but we don't see that when we look at the genomic context. But it's impossible to tease apart these things because sequences are not what evolution acts upon. Evolution acts on function, and function is a property of proteins and not sequences, when you think about that. Thus, by only focusing on sequences, we miss critical context, without which we treat every variant equal by design, even when using single amino acid variants. So clearly you need to incorporate structural insights into these proteomes you study in the environment, but it's extremely difficult because, as you know, protein structures have been extremely difficult to predict. But while we were thinking about these problems and using some archaic methods to do that, something amazing happened. AlphaFold came out. And we were able to propose an updated workflow to this using protein predictions from AlphaFold. It's staggeringly simple, embarrassingly simple almost, but this is how it goes. You have an operating frame and a reference sequence. Now you can predict the structure for it. And then you can also predict the ligand binding residue in this particular protein, which is the active site of the protein that's extremely critical for its function. Then you can resolve intrapopulation genetic variants in the context of predict protein structures and ligand binding residues to be able to interpret genetic variation in a structure-informed way. So now you see that all these similar looking variants perhaps occupy very distinct parts of the protein. For instance, some variants are going to be only single codon variants and will never yield any single amino acid variants. So there are no synonymous, non-synonymous polymorphisms, perhaps. In another case, perhaps you will observe the single amino acid variants are largely occurring in high relative solvent accessible residues of the protein. And perhaps in other cases, you will realize that some single amino acid variants occurring right next to the ligand binding site that that indicates that perhaps there is something going on here. So this residue that is variable in the environment is not same as this residue, because if you are inside the protein, you are more likely influence its folding and function. So biochemists knew this forever, that solvent accessible parts of proteins are more likely to accumulate variants, but we have not been able to include that in our thinking very much because the sequence space would not enable us to recognize those kinds of features of proteins. So then we asked with Evan Kiefel, how did synonymous and non-synonymous polymorphism distribute through SAR11 proteins? And joined the effort Özcan as a former programmer in my group. And then what we did, we took all the metagenomes, all the variants, and we predicted 800 SAR11 structures and overlaid them on these structures. So I'll show you a very quick example. This is a nicely cherry-picked example, of course, because I love this example so much. This is the hydroxyl methyltransferase gene in SAR11, which binds to serine for a very critical metabolic function. And what you see in the center, the blue part is where the protein is bound to serine. And even in this preliminary view, actually, you can see how the variable amino acids we find in the environment are far from the ligand binding site, because now we can talk about the fact that this protein cannot afford to have any variance closer to its critical site to maintain its function. So let me put it this way so it's easier to see. The same display I'm just showing from its particular axis that maximizes the visibility of the variance. As you can see, they're all at the outskirts of the protein, basically. So these are non-synonymous variants. These are single amino acid variants, but they are not necessarily as non-synonymous as one would expect. They are very precisely avoiding particular parts of the protein. So one could, in fact, say that the insides of the protein is under very heavy purifying selection, and outsides are not necessarily as much. Therefore, even these non-synonymous variants are somewhat synonymous. So you can now expand the universe of our thinking regarding how variants contextualize in these genomes. So what I'm showing you here is a very clear example, and yet there is also a lot of signal for the rest of the genes. For instance, when we look at how synonymous and non-synonymous variants, as we calculate much more effectively, distribute across relevant proteins as a function of their distances to ligand binding sites in angstroms here. This is what we see if we were to randomly shuffle everything. So this is the null distribution, random shuffling of potential variants and their distance to ligands in proteins. And this here shows synonymous variants. It almost perfectly follows, not quite, but almost perfectly follows the distribution, and which means synonymous variants can occur pretty much anywhere. Yet, when you look at the non-synonymous variation, you see a significant shift in this curve. And when you, what this means is the variants that are non-synonymous are trying to avoid the residues that are buried or closer to the ligand binding site. And this is true for relative solvent accessibility. You see, again, a significant shift in these curves. So comparing these two metrics also is very illuminating with this beautiful visualization Evan Kiefel came out with. You see how there is no structure to see when you compare how distance to ligand binding site and relative solvent accessibility play out in the context of protein given synonymous variation overall in the genome. But look at this, this is very different. Non-synonymous variation truly respects both of those aspects of proteins. So what I'm showing you essentially tells us that non-synonymous variants are maintained rigorously. They generally avoid residues that are closer to ligand binding sites. They generally avoid residues that with low relative solvent accessibility. So essentially, also, let me finish with this. Synonymous variants don't seem to be much of a concern in comparison, but actually this is not true. Because while the general trend suggests that, in fact, the reality is much more subtle than this. And we have a preprint on this, which is currently in review. If you'd like to take a look at that, please feel free to do so, or write to me. I'll send it to you later. But what I want to show you is I want to go back to this figure and talk about the fact that not all non-synonymous variants are further and further away from the ligand binding residues. As you can see, there are some that are right next to those actually. So now when you recognize, when you're able to bring together environmental variants along with structures, and when you realize the general pattern that suggests non-synonymous variants are escaping these regions of the genes, then you are more likely to remove all variants that are most likely under neutral processes, maintained by neutral processes. But then when you find some that are not, then you get closer to understanding perhaps some evidence for adaptive processes or lack of purifying selection, depending on environmental conditions. But this way you have much bigger grasp on the likely evolutionary processes that act on individual environmental population as a function of change in the environment. So I want to stop here because I want us to have some time for discussions. Therefore, I'm not going to talk about the future and translation regulation, metabolism, and so on. But we have been developing the software platform to put all these ideas in. So essentially, you can get very comprehensive data from marine environments using microbial genomes and metagenomics to be able to link them to protein structures. Every single figure I showed you today, including the 3D visualization of proteins and their variants, are coming from this platform. So in case you are interested, I'd like you to know that there is an open source platform out there for that. And the other thing I want you to consider for a second is the complexity of the data that I've been showing you. Not only the complexity of single amino acid variants and single codon variants, but also the complexity of in the size of metagenomes and inclusion of a structural thinking into this problem to address some of the fundamental questions of evolution at the interface of populations where environmental change is a significant concern. So data scientists, those who have skills in computer science or programming, of course, have a huge advantage to address some of these problems. So if you ever consider these things, yet if you have questions and concerns and would like to bounce some ideas from someone who has been studying these things, please feel free to reach out to me again. And I'd like to finish my talk by thanking my colleagues who, until recently, I was at the University of Chicago, as Anna mentioned. And I'm very thankful for these people and their contributions to our efforts, as well as Simon's Foundation and others who contribute to our research. But also, I want to thank some of the new members of my group in Germany who are not listed here, such as Mete, Alex, and Cynthia, who are just working with me in Germany to go after similarly interesting questions, at least in my opinion. And I would like to thank you for your time and your attention. And with that, I'm going to stop sharing my screen so we can see each other. And I'm happy to address any question you may have.
Thank you very much, Mary, for this fascinating talk. I'm going to stop the recording